Hey y'all, today we're going to talk a little bit about cell theory. And so we're going to talk briefly about different types of microscopes um, and the three tenets to the cell theory as we currently understand it. So we should probably get started with talking about the individuals here that are involved with this whole process. So one of the very first guys that we have to give credit to is Anton van Leeuwenhoek from the 1600s. We give him credit for having the first microscope, although it wasn't very complex. He looked at pond water and he saw a whole bunch of little creepy crawlies moving around in this pond water, and it was pretty important. He called them wee beasties. And it was really significant because prior to this, water was considered clean from a religious and a practical standpoint. The fact that Leeuwenhoek found things living and moving in it was really disturbing to a lot of people. Kind of a gross FYI on the side, I mean, he was really interested in microbiology. So when he got the runs one day, he decided to put that under the microscope and saw some of the same types of wee little beasties that he saw in the drinking water. Now, we don't actually give him credit as being the first guy involved with the germ theory, but his early work did start the ball rolling for the scientists that are involved in germ theory. So obviously the really close microscope images here are not going to be what they were able to see, but you can see some pen and ink sketches that were made by early scientists. Robert Hooke gets credit for naming cells, so he's considered the father of cells. And we can see his version of a microscope here. He observed cork under the microscope in the 1600s. And he saw these tiny, tiny little chambers, he called them. They were right next to each other. He called them cells because they reminded him of monk rooms in a monastery, and they're called cells. So that's where he got the name. And what he actually saw was the cell wall of the plant cells because they had already died. All he saw was the leftover carbohydrate-based cell walls. And these are a couple of his images. So you can see the sketches that he made of cell walls, and then you could see this very close-up of what looks to be a flea or a louse of some sort. Robert Brown in the 1830s, so this is almost 200 years later, observed that some cells have a dark structure within them, specifically plant cells. He observed the nucleus. Matthias Schleiden in 1838 was looking at plant cells, so he was a botanist, and he looked at a lot of different types of plant cells under the microscopes, and he observed that plants are all made of cells. Theodor Schwann in 1839 observed the same thing about animals. So he looked at a lot of different animal tissues under the microscope, and he saw that animals were also made of cells, which was pretty important. Now, the way that I keep Schleiden and Schwann separate in my head is that Schwann sounds like swan, which is an animal. And baseball players Schleiden slide on the grass, which is a plant. Or softball players. Rudolf Virchow in 1855 actually observed cell division. So he stated that cells come from pre-existing cells. They arrive from the division of other cells. So this was kind of a big deal. Prior to this time, people believed in a lot of different things, and they actually thought that this spontaneous generation concept could happen, where if you, I don't know, left food out, all of a sudden bad things would grow. Not because something was already in there or something was in the air or something was transported by bugs, just spontaneously it would grow. And so Virchow's work was really instrumental in this concept that we're looking at the growth within that cells are coming from cells. So there had to be something that started that process. Okay, so at this point, you just need to make sure that you've got your timeline. It needs to be roughly, roughly measured out so that the 1600s are together. You got your gap and you got your 1800s together, because we're really looking at the fact that we have a little bit of knowledge and then a big gap of time and then some more knowledge and then some time and then us, right? So we want to make sure that our timeline is roughly reflective of that gap. Thanks for studying with us.